Buenos días.
Good morning, buenos dias, and welcome to everyone who's here in the room and also online. My name is Laura Lewis. I'm the director of the Genetic Resources Program, and I'm honored to welcome Blake Vinenhept here from Montana State University, Department of Microbiology and Cell Biology. We're really excited to hear his talk on CRISPR defense to crop protection. And it's an exciting opportunity for us to hear about work from one of our collaborators. So I'd like to welcome Blake to be able to come and share with us about his research and how it can be applied with the work that we do here at CIMMYT. Thank you.
Good morning, all. So first, I'd like to introduce Blake. Blake is a friend and a scientist in the area of gene editing. So he's a professor in microbiology and cell biology at Montana State University, and Winifred Absorption's plant science endowed chair. His research focuses on the molecular mechanism of the microbial immune system, including CRISPR, and how this system can be harnessed as powerful tools for biotechnology and genome engineering. And during his postdoc, he worked with Jennifer Doudna, Nobel laureate from UC Berkeley, and he worked during the early days of CRISPR-Cas9 invention and downstream applications. Now he is quite a bit involved in plant and agriculture. So he uses his new tools and technologies for agricultural development. And he has received many prizes, NIH Director's Early Career Scientist Award, Presidential Early Career Award for Scientists and Engineers, etc. So we welcome Blake. I welcome Blake. So Blake, please, the stage is yours.
Good morning, everyone. Thank you, Nadia. It's really a pleasure to be here. It's quite an honor. First time to Mexico City, first time to CIMMYT. And although, we're emerging or trying to get a little bit more involved in plants and wheat in particular, that's not been the focus of my research historically. As India mentioned, we have a long history of studying microbial immune systems, which I don't think are biological idiosyncrasies. What we're beginning to learn is that the immune responses in microbes cross domains of life. What's true in elephants may really be true in E. coli, but it's also true. What's true in E. coli might be true in plants and might be true in humans. So today, what I want to do is start by telling you a little bit about the work on microbial immune systems that started just down the road from Montana State University in Yellowstone National Park, in the boiling acid hot springs of Yellowstone National Park, and how that really led me to the work that we're doing now. Oh, there we go. And a part of the lab that's trying to transition to moving to studying pests in plants, but in wheat in particular. As some of you may know, Montana is the fourth largest state in the United States, but there's only 1 million people total in the entire state. And much of Montana is dedicated to wheat production. So this is a big wheat production state. And I'll tell you a little bit about the major pests that we've been challenged to try to address. But before I do that, I just want to give you a quick introduction to some of our work on the microbial immune response. And my work as a PhD student was to run, hike, bike, ski around Yellowstone National Park. It's a beautiful place. It's full of wildlife, but also some natural beauty that comes in the form of microbes that live in these boiling acid hot springs. And my job was to scoop up bits of boiling acid, bring it back to the lab, and see what we could grow. And more times than not, what we grew was an organism called Sophilobus solfataricus. It's an aerobic heterotroph that grows at 70 degrees Celsius and pH 3. And nearly every time we sampled these hot springs, we could always come back and find new viruses. Sometimes these were viruses that had never been seen previously. And of course, you do that a certain number of times and you start to wonder, how is it that these microbes can thrive in these habitats under the selective pressure or invasion of these infectious agents? And that led me to an early interest in CRISPRs. This is a CRISPR complex that doesn't come from Sophilobus, so this is a little misleading. But the gist of it is that, as most of you know, CRISPRs are adaptive immune systems in microbes, and they work by an RNA-guided the RNA here shown in red, RNA-guided defense that recognizes double-stranded DNA and cleaves viral DNA in a sequence-specific fashion. But just like any tit for tat in molecular biology, viruses, of course, have to face this new selective pressure from these immune systems. And doing so, they evolved anti-CRISPRs, or basically small proteins that function as molecular wedges that stick inside the CRISPR complex and block the CRISPR complex from binding to the target DNA. And these anti-CRISPRs have also been implemented in a variety of different biotechnological tools or implementations. But CRISPRs don't just target DNA. In this conflict over time, CRISPRs have evolved many different forms and functions. And one of these different systems, a so-called type 3 system, I think most of you would be familiar with Cas9 or Cas12 for the genome engineering applications. But more recently, a system called the type 3 system was discovered. And these type 3 systems don't target DNA at all. They exclusively target RNA. And they cleave the target RNA. And in fact, what we've shown is that just like Cas9, you can port these into human cells. And when you put them in human cells, without any PAM restrictions or guide design rules, you can target any transcript. And what results primarily is 95 to 99% knockdown without concern of where you target the RNA. So these are highly efficient, much more efficient than something like RNAi, for example. So one of the questions that we have in collaboration with Anindya's group is to ask, can we move some of these RNA-guided, RNA-targeting CRISPR systems into plants in a way that we could uniquely target parasites that don't have a DNA intermediate in their life? For example, RNA viruses that infect different plants. In addition to the work that we're doing within India, we've also been challenged to address something called the wheat stem sawfly, which isn't a fly at all. It's actually a wasp, but I'm not an entomologist, so I wouldn't know the difference if I'm honest. What I do know is that these insects grab onto the stem of a wheat, they inject their eggs. These eggs then mature into larvae that burrow down through the stem of the wheat, compromising the integrity of the stem as they grow. When they get about 6 inches off the ground, they use their feces to create a little bit of a cocoon where they undergo a 90-day metamorphosis, oftentimes through a Montana winter, which can be minus 50 degrees Celsius. So this is pretty bizarre biology for somebody who studies microbes. And it's been a pretty exciting new experimental direction for us, as we're now bringing in insects into the lab. This is the first time in my lab where we actually have dirt and plants and stems. And in fact, what we've recently done is developed, I don't know if that's the video's working or not, but I can't get to it on my own screen. The gist of it is that we've now taken these stems out of the fields, cut them open, taken the larva that hasn't started to develop yet, created an artificial stem in the lab, and now we can watch these insects go through their entire developmental process from a larva into a fully fledged insect. And this is a powerful system because now we can test new insecticides in rapid order on a larger scale before we go into trying to engineer plants to say try to develop a new biological insecticide that might work against these insects. All right. So now I'm going to shift gears a little bit and tell you about some of our work on microbial immune systems. And I understand that this might not be the wheelhouse of everybody in the room. So I'll try to explain at a little bit higher level how we think about these things, how we've discovered them, and what we're learning about mechanism and how it might apply beyond just microbes. So I already told you that we had been working on CRISPR systems. And that work led us to anti-CRISPRs. And this conflict between CRISPR and anti-CRISPR drives diversification. That's true for all of biology, whether it's SARS coronaviruses that infect humans and this tit for tat that happens between the viral genome, which evolves over time, and your own immune system, or happens inside of a microbial cell. It drives diversification. And we could have gone on studying CRISPR, anti-CRISPR for probably the rest of my career. But I was lucky enough to get a call from a friend of mine at the Pasteur Institute in Paris. And he said, hey, we recently discovered a new immune system that they ironically, or maybe apropos, they called it Paris. And this Paris system, which we determine the structure of, and you're seeing here, which looks something like a propeller, doesn't recognize foreign DNA like CRISPR's at all. Instead, more like some of the components of your own immune system, this system recognizes foreign proteins, specific foreign proteins. And when this complex recognizes an anti-CRISPR, that drives a conformational change in an ATP-dependent fashion that ejects one of the blades off of this propeller-shaped complex. And these blades of this propeller-shaped complex then go on to, sorry, there's a bit of a delay here, go on to cleave tRNAs in the cell. If the tRNAs, that's what's being shown here, 30 minutes after activation of this Paris complex and release of this yellow subunit off of this propeller-shaped complex results in the degradation of tRNAs, in this case specifically tRNA lysine. If you eliminate all the tRNA lysines in the cell, that triggers translational arrest, and without translation, no virus can survive. So that's how it shuts down the viral infection. So what I just described is an ATP-dependent antigen sensor that releases a toxin that cleaves tRNAs in the cells and shuts down translation in the host and thereby prevents infection. All right, so this was just a jumping off point for us. We didn't know that at the time, of course, but it got us to thinking, if this one system called Paris relies on an ATPase, an ATP-driven antigen sensor, then is it possible that this platform is common to a lot of different immune systems? So to address that question, Murat Bukarik, a postdoc in my lab, took a series of sequences related to this ATPase, created a profile hidden Markov model, queried a custom database that we maintain in the lab composed of all open reading frames from all bacterial and archaeal genomes, and then created a phylogenetic tree. And that's what you're seeing here. But then he took this one step further, and every tip on this tree is then flanked by one of these lines, which represents the amino acid for that particular protein from N terminus to C terminus. And you see that this line in every case for every member of this tree has this different color scheme, which is colored according to the conserved domains of this protein. Nucleotide binding domain 1, these two insertion sequences which are involved in oligomerization of this complex, and then nucleotide binding domain 2, these two NBDs, so-called NBDs or nucleotide binding domains, are necessary for turning over ATP. So they're a fundamental component of all of these kinds of proteins. But if you look just downstream in the genome of all these organisms, wherever you find one of these REA or these ATPases, you find a family of proteins that have a top rim domain that's associated with this nuclease activity. So this is consistent with what I just showed you, that we have this ATPase sensor in the middle, and then just adjacent, this is one of the benefits of studying prokaryotes, genes are clustered in functional organization. So if you find a gene, the gene next to it is probably related to it in terms of function. And that's exactly true here. You find the sensor, and then just downstream, you find the nuclease, and they form this complex. But what caught our attention is that if you look down this tree, you find not just the parasystems that have the ATPase and the nuclease domain, but rather you find this ATPase that's now not flanked by the top rim nuclease, but rather an HNH nuclease, and then looking upstream, a reverse transcriptase. So some of you might be familiar with reverse transcriptases. There was a Nobel Prize awarded for the discovery of reverse transcriptases to Howard Temin and David Baltimore in 1975, and it led to the discovery of RT inhibitors, or reverse transcriptase inhibitors, that now 30 million people in the world take as a consequence of the largest pandemic on the planet, HIV. So most people thought that reverse transcriptases were part of retroviruses or parts of viruses that were necessary for the replication and spread of these viruses. But what I'm about to tell you is that reverse transcriptases are prevalent in bacterial and archaeal genomes, and there they do the exact opposite. They fight viral infections rather than propagate viral infections, and I'm going to tell you how they do that. So these systems that I'm going to tell you about right now are so-called retrons. They've been studied or known about for quite some time, but their function remained elusive. Here's just a schematic of the operon. I told you that genes of related function cluster on the genome. So here you see that centrally encoded ATPase that's flanked upstream by this reverse transcriptase, downstream by this H and H, or this nuclease. When I say H and H, you might think Cas9. H and H domain is part of the Cas9 nuclease, but here it's doing something very different. All right, so I told you that retrons had been discovered and known about for quite some time. And in fact, in the 80s, in total DNA purifications of Myxococcus xanthus, you could see that there's this strong band of extra chromosomal DNA and total DNA preps from this microbe. Nobody knew what this was or where it's coming from or why, but it turns out that this DNA is being made by the reverse transcriptase here, and it's using an upstream non-coding RNA as a template to make that DNA. Nobody knew why, but this was happening, and you could clearly see it in these total DNA preps. The way that it works is quite surprising as well. This is RNA, so you have a 2' hydroxyl on the sugar, and this branching guanosine serves as a primer. This 2' hydroxyl on this branching guanosine serves as a primer for the reverse transcriptase, and the reverse transcriptase uses a segment of this non-coding RNA as a template to make DNA. And in doing so, it creates a covalent RNA DNA chimeric molecule. It's both RNA and DNA now. But the function, again, of this chimeric RNA DNA remained enigmatic until 2020, when Rotem Sorik's lab at the Weizmann Institute performed this screen against viruses. And what they showed was that these retrons that had been proposed to have many different functions, none of which turned out to be true, are actually involved in viral defense. And what you're seeing here is just results from a plaque assay showing that this particular retron system, the so-called EC78, provides high defense against T5, but not these other viruses. And then these other immune systems provide defense against these other viruses, but not others. All right, so this is where we got involved, and we wanted to ask, okay, so these are viral defense systems, but how do they do that? Much in the same way that people discovered CRISPRs fight off viral infections, but nobody knew how until we realized that they're RNA-guided DNA cleaving machines. And then once we understood how, then they could be creatively repurposed. So if you come up with a creative repurposing here, it could be a big deal, and please share it with me. All right, so that was a joke. I don't really expect you to share it with me, but if you have some creative ideas, I'd love to chat about it. All right, so what we did was took this immune system out of the natural organism, cloned it on a plasmid, put it into E. coli, and Jaron and Royce, 2 postdocs in my lab, asked, does it provide defense? And this had been seen before, but I'm just going to share how these experiments work for anybody who's unfamiliar. You can see that the virus concentration is highest on this end and lowest on this end. These are lawns of E. coli that T3 virus is being spotted. So here you see a large plaque, and the plaques get smaller as you go to higher and higher dilution. This EV stands for empty vector, meaning no immune system. This is an immunocompromised strain of E. coli. But if you put this plasmid in this cell and now express the retron system, you can see that there's considerable level of defense. In fact, let's just count it. 1, 2, 3, 4, 5, maybe 6 logs of defense against this particular phage. Similarly for T5, to a lesser degree, but still 1, 2, 3, maybe 4 logs of defense against T5 when you're expressing the retron system versus an empty vector control. So we overexpressed the system, used affinity tags to pull down and purify the complex, almost a 400 kilodalton complex. If you look at an SDS page of this peak, you can see that all of the protein components are present, the ATPase, the reverse transcriptase, and the H and H. And if you run a denaturing page of the nucleic acids, you can see that both the DNA and the RNA are present. So this holo complex is intact. One of the things that we do in my lab is use cryo-electron microscopy to look at complexes like this, to understand the molecular blueprint of these machines, with the idea that once you have the molecular blueprint, you can understand how they work. We were able to determine 3 structures of this complex, but even before structure determination, we could see in these 2D class averages these prominent protuberances that stick away from the complex. This is the DNA that's made by the reverse transcriptase using the upstream RNA as a template to make this DNA harpoon-like feature that pierces the center of the complex. This harpoon is cradled by 4 molecules of the ATPase. Here you can see that kind of have the DNA in a bear hug. And then the reverse transcriptase is perched at the top of this complex. This complex in particular, this structure, doesn't have an H and H associated with it. We think that the H and H can fall off and on, so it's a little bit volatile. But here you can see the H and H is in the down position and here in the up position, which made the structure determination slightly more complicated, but revealed some dynamics that would have gone probably undetected using other methods previously. All right, so they were all around 3 angstrom resolution reconstructions. So what I've shown you so far is that retrons are immune systems and that they form these unusual complexes by using a reverse transcriptase that uses a non-coding RNA to make a DNA harpoon that serves as a scaffold for the ordered assembly for these ATPase molecules, 4 molecules in total, that wrap around this DNA. And then this scaffold then recruits a toxin, an H&H nuclease, that keeps it safely sequestered away from doing any damage, until which time it needs to be released and provide the cell from protection. But how does that happen? What I'm going to tell you is that this system senses a virally encoded nuclease, and once that nuclease is released by the viral infection, the nuclease destroys this DNA harpoon, which disassembles the complex and releases the toxin, which then shuts down the cell from replication and prevents viral propagation. And the way that we found that was we kept challenging these cells that are expressing the retron system with new virus. And we selected for virus that was insensitive to the CRISPR system, to the retron system. So viruses that could grow even though the retron system was there. And then we sequenced these mutants, mutant 1, 2, and 3, and we asked what mutations are common between all viruses that are able to escape elimination by this immune system. And I won't go through each of the different examples, but what we honed in on was this virally encoded exonuclease called the T3 exo. And what you're seeing here is not a plaque assay at all, but rather a dilution assay of E. coli. So this is just the highest concentration of E. coli, tenfold less, to the point where you start to see not a lawn where it was spotted, but rather individual colonies. If you express the exo, it's not toxic at all. But if you express the exonuclease from the virus along with the retron system, now you see several orders of magnitude of toxicity as a consequence of expression of that exo. So we could do this in cells where we express the retron in both of these lanes and then either don't express the exo or express the exo. And what you see here is that the DNA created by the reverse transcriptase is eliminated when you have the virally encoded exo, supporting this model where the exonuclease from the virus destroys this DNA harpoon, which functions as a molecular sensor for viral infection. Once this gets degraded by the virus encoded nuclease, then that releases the toxin, the H&H, which then shuts down the infection. But the next question is, of course, what's the toxin actually doing? And I've kind of given it away in this cartoon already. But surprisingly, this H&H nuclease, what it's doing was a little unexpected. It's destroying tRNAs. So I won't lead you through all of the experimental details that led us to this notion of translational arrest, but we had some clues along the way that translation was being stalled as a consequence of infection. And in fact, when we deep sequenced all the tRNAs from the cell, we saw selective depletion of all 4 codons for serine. So without serine tRNA codons, of course, you can't replicate. There can be no translation. Shuts down translation, leads to translational arrest and viral protection. So that's the mechanism of defense of this system. So I'm just going to recap a little bit before going into the next chapter. What I think I've told you so far is that we started studying CRISPRs, and that led us to this new system called Paris. And the reason that we started studying Paris is the anti-CRISPRs that we were studying before are sensed by this Paris system. This is an ATP-driven protein sensor. And when it senses an invading viral protein, it releases a nuclease colored in yellow here that then destroys this tRNA, resulting in translational arrest. That got us wondering, is it possible that these ATP-driven protein sensors aren't unique to Paris, but rather more broadly distributed? That bioinformatic experiment led us to retrons. The retrons led us to the determination of this structure of this retron complex that uses a DNA sensor and scaffold to hold this complex together and sequester this H&H nuclease, which is part of Cas9, but in this case, is part of an entirely different complex that doesn't cleave DNA at all. It cleaves RNA. In fact, it cleaves tRNAs in much the same way that this nuclease cleaves tRNAs and shuts down translation in an infected cell. But by this time, you're probably starting to figure out the model in my lab. Now we've got interested in RTs. So we were initially interested in CRISPRs. That led us to ATPases. We started looking around at different ATPases, and now we've noticed that these RTs are playing an important role in immune systems. So Murat now created a profile hidden Markov model of of RTs, and asked, what's the phylogenetic distribution or diversity of reverse transcriptases in all prokaryotes? And what he found was there's quite a bit of diversity. We were studying just a small clade here called retrons. And the next question became, what do these other systems do? So in collaboration with Sam Sternberg's lab at Columbia, we went after this system that at the time was called UG28. This is a clade UG28, unknown group 28. There's more than 28 different unknown groups. It's just a clade that nobody knew what it did. So we thought, oh, this is a landscape that's unpioneered. Let's go look around there and see what we can find. And what we found was a family of immune systems that include only a reverse transcriptase. My screen is blocked a little bit here, so I'll have to just do the best I can, but that red arrow points to the operon structure here. The DRT9 gene is flanked upstream only by a non-coding RNA. So unlike the retrons, where you have a reverse transcriptase, an ATPase, and an H&H nuclease, and a non-coding RNA, this is a much simpler system. Only 2 components, a non-coding RNA and 1 protein. This protein, it turns out, assembles with the non-coding RNA in a 1-to-1 stoichiometry. So that protein, reverse transcriptase, and the non-coding RNA interact with each other. And then that protein RNA complex self-assembles into a homohexameric complex. You see 1 trimer of it, which there, the RNA forms something of a crown around the top of this complex. 1 segment, highly conserved segment of the RNA is positioned in the active site, and it serves as a template to make poly A. So in every axis of this threefold axis of this complex, this machine spews out poly A sequence. And what that does in response to a viral infection is shut down the infection in a way that we think involves the sequestration of single-stranded DNA binding protein. But I'll be honest, the mechanism of defense still remains slightly enigmatic. That's something that we continue to pursue. But I'm not going to tell you about the gory details of this system because I want to tell you about this other system. And the reason I want to tell you about this is I think it changes how we think about biology, really fundamental change in the central dogma of biology. And I'm going to try to defend that statement. But these systems are called DRT2s. They're not very far from the system that I just told you in terms of their phylogenetic position. They're a neighbor, a close neighbor. But what I'm going to show you is they work very, very differently. So most of you are familiar with the central dogma of biology. And this was something that was first articulated by Francis Crick in 1958, in fact, at a symposium. But this figure that you might be familiar with wasn't formalized until this paper that was published in 1970 in Nature. And this paper was published in response to something I told you about earlier, the Nobel Prize for reverse transcriptases, because at the time it was a bit controversial, saying that reverse transcriptases had changed the central dogma. So Francis Crick wrote this editorial, or this piece in Nature, to say, no, it doesn't change how I imagine the central dogma. It just adds more information to what I already conceived. So what he was trying to understand at the time, of course, was how genetic information flows from DNA to RNA to protein. But we already knew that DNA could replicate. That's part of every cell, but also part of every DNA virus. And what became clear, of course, is that RNA viruses could also replicate. But what started the controversy and what I mentioned previously was the discovery of reverse transcriptases, which could reverse the flow of genetic information such that it wasn't unidirectional from DNA to RNA to protein, but rather from RNA back to DNA. But what's so exciting about the discovery of these DRT2 systems in prokaryotic genomes is that they do something entirely different. They take DNA, a non-coding RNA that's encoded in the DNA genome. So we go from the DNA genome to a non-coding RNA in this case, and then DRT2s use this non-coding RNA to make a new DNA that's not encoded anywhere. It's not in the genome. It's not in the RNA itself. It's a new piece of DNA that's extra chromosomal to the genome. And it turns out that that DNA has the information to activate transcription, which results in a new RNA that's also not found anywhere else. And that new RNA has all the information necessary to be translated into a new protein that you would have never discovered by just looking at the genome. So I'm going to tell you a little bit about how the system works, but I think what it reveals is that there's a new dimension in the genetic code that had never been previously appreciated. So spoiler alert, like part of the reason that we're interested in these systems is we wanted to ask, is this phenomena perhaps more broadly applicable than has been appreciated? And if so, then it suggests that there's a bunch of proteins with novel functions that we're not even considering. So I decided to go all in on DRT2s and to really start understanding how they work and how we might be able to repurpose them. So I'm going to give you a little bit of detail here. These systems are a bit complicated, so I'll try to go slow and yell if you are not following me. But what you're seeing here in this top line is a zoom in of this operon where we have the reverse transcriptase in this purple arrow, and this YXDD is the active site of all reverse transcriptases. Upstream of this reverse transcriptase, you see the architecture of this non-coding RNA with the transcriptional promoter just upstream of the non-coding RNA. This transcript makes a non-coding RNA that's highly structured. It assembles into this predicted 2D structure that includes a 5' scaffold and a 3' scaffold. But in the middle here is the sequence that will serve as the template, or what we call a cargo, with the hope that we can replace this with any sequence by design. Turns out to be harder than it sounds, but that's our goal. And what we know is that this sequence recruits the reverse transcriptase, and I'm showing this part of the RNA now here in just a linear representation, the 5' end, then you get to this so-called template switching motif here, then the -35 on the 5' end, through this section, which is about 95 nucleotides, then to the -10. You see that here in this linear representation, the rest of the cargo, and then the template switching motif. So this is the template for the reverse transcriptase. So it hops on to this RNA and it starts transcription at this position here. Then it goes around one loop of this template, creating a cDNA, moving along the template in a 3' to 5' direction, going in this 5' to 3' polymerization, resulting in the -10 and the -35. But that's not biologically meaningful at all, right? It's not until the reverse transcriptase hops templates and bridges across this junction to then start polymerization again in a new location, not where it started the first time. And now it makes another lap around this template. And as a consequence of that, it moves the -35 and the -10 into position such that they're separated by 17 nucleotides, which is an absolutely critical distance necessary for a bacterial sigma factor to activate transcription. So you only get this promoter as a consequence of template hopping and doing another loop around this RNA. So this happens over and over and over again. It's not just once or twice. This happens ad infinitum. We don't know how long they are, but they're thousands of base pairs. They create a bit of a smear on a southern blot. So the distribution is a little bit heterogeneous, but let's say it centers around 1,000 nucleotides. In a way that we don't understand yet, the second strand of DNA gets made, and collectively, this results in an mRNA that is then translated, and this is called NEO, for nearly endless ORF, for the reasons that I just explained. It's nearly endless, and in theory could go ad infinitum. There is no stop codon encoded within this sequence. All right. What we knew about it at the time that we got involved is that if you made this protein, just ordered clone by phone, order this sequence and express it in bacteria, with 2 repeats, you see a dramatic cutoff or a stall in bacterial growth upon induction. So here's just a control, bacterial cells growing just fine. Then you induce GFP. When they're growing while expressing GFP, there's very little lag on the growth of the cells. They grow just fine. You do this, you replace the GFP with one of these repeats of these proteins. It's only about 24 amino acids. There's very little toxicity, but if you do it with 2 of them back to back, this results in toxicity or at least plateau of cell growth. And if you do it with 3, the phenotype's even more profound. So the big question, of course, is what are they doing and how are they doing it? And the other big question is it only one sequence that's capable of this, or are there many? You can see that there are many. In fact, Murat found about 1,800 of these sequences. And the system that had been studied before was restricted primarily to one clade. So I showed you this kind of analysis previously, but just to recap, this is a representation of the reverse transcriptase with the active site, the YADD active site. And if you look just upstream, you see this non-coding RNA. This is the 3' scaffold, 5' scaffold. This is the promoter. Here you have the template switching motif, -35, -10, and this is the template. This is the cargo. It turns out that this cargo is very diverse. And if you look in different systems, it's not just this ACA template switching motif. These switching motifs can vary dramatically. So if you're going to look for new systems and new genomes and perhaps across domains of life, you can't just look for the ACA because it varies depending on which clade you're in. But one thing that's conserved is that when it does a lap around the second lap around the template, that results in a spacing between the -10 and -35 that reconstitutes the transcriptional promoter. That seems to be a feature that's highly conserved, and it was pretty gratifying, or at least verified, that some of our predictions have merit, because we're just sequence gazing at this point. We don't know if these predictions of where the template starts and stops are really true, but since they reconstitute the -10, -35, that seems to verify that they're probably legitimate cargoes. The other thing that verified that they're legitimate cargoes is that 98% of our predictions are all divisible by 3. That means that they're probably all resulting in neoproteins, but the proteins themselves are highly diverse. So the big question then becomes, if these are highly diverse proteins, do they all result in the same phenotype? So we cloned from each of these different clades 2 or 3 examples of them, and then expressed them in E. coli, and then measured the rate of the growth rate of the cells after induction. And what you can see is that if you do this with GFP, the cells have a high growth rate, and there's very little lag on growth as a consequence of expressing GFP. If you do this with the positive control that was discovered by Feng Zheng and Sam Sternberg's labs, you can see that there's a slight negative growth rate resulting in this toxicity that I described earlier. If you scramble that sequence, then there's no toxicity. So the sequence itself and the order of amino acids that's encoded by these cargoes, it matters. All right, so then we go to all of our experimental systems, and without exception, they all phenocopy this system that had been previously studied, suggesting that they all have the same phenotype, stalling or stopping the growth of bacterial cells. So we wanted to ask, how is this actually happening? And we got some clues that there was some depolarization of the cellular membrane. And so we ask, what happens to ATP concentrations when you start to express NEO? And the result that I'm showing you here is a dramatic decrease in ATP concentrations as a consequence of NEO expression. And this mirrors what you would expect in the growth curves. The cells stop growing, and the reason that they stop growing is that the membrane is depolarized and that there's no ATP in the cells. We tested this using a variety of different techniques, using some reagents from Thermo Fisher for testing membrane polarization, followed by back sorting. And what we found is that only when you induce the expression of NEO or a positive control do you get membrane depolarization. So the big question was, why hadn't this been observed before? And we well, one reason is that nobody looked. And the other reason is that the membrane aspect of this protein is not immediately apparent. In fact, if you do transmembrane predictions with most of the available software, you don't see any evidence of a transmembrane domain. But if you use one of the slightly older programs and then you start to increase the number of repeats, it's only at this intersection between repeat 1 and repeat number 2 that you start to get a really strong signal for a transmembrane domain. So we asked, what if in silico we just mutate those residues that are thought to be important for making the transmembrane domain and then repeat the prediction? And in that case, you get no predicted transmembrane domain. And if you do it in both the N-terminal and this mid transmembrane domain region, it just flat lines. There is no prediction for transmembrane domain. So then what you're seeing over here is the AlphaFold prediction of this peptide, this neoprotein, and we're zoning in on these residues that were thought to be critical for the transmembrane activity. Then we made site-directed mutants of these specific residues and asked, does that eliminate the ability of this protein to kill cells? And those results are what you see down here. Normally, growing cells Now you induce wild type neo-expression, results in a flatlining of cell growth. If you make mutations in just repeat number 2, the phenotype is slightly eliminated. And if you make mutations in both, the cells grow much, much better, suggesting that these few amino acids that are necessary for transmembrane function are actually critical to neotoxicity. In addition, we went and looked inside the cells. So this is just a Z-stack through E. coli cells that are expressing a GFP tagged neoprotein. And what you can see is that GFP itself is primarily localized to the cytoplasm. But if you GFP tag neo, that brings it largely to the membrane. And in fact, you can see these distinct puncta that are localizing here. You still see a few puncta in the transmembrane mutant, but a majority of the signal is cytoplasmic rather than membrane associated now, suggesting that you've disrupted the membrane association, which is consistent with the loss of the phenotype that I showed you in the previous slide. I think in the interest of time, I won't describe this, but the gist of it is that this is quantification of what we see in these images, where we do a transect of the signal, GFP signal, across the cell. So if you measure the GFP across this cell, what you see in a GFP expressing cell is you have high expression of the signal, and most of it is in the cytoplasm. But if you do that with the EGFP tagged neo, you have much lower signal, which is to be expected when you're expressing a toxin, right? You get a lot less protein expression. But now most of the signal comes from the fringes and less signal. You get this concave shape because you have most signal aggregating on left and right side of the cell, here and here, with a lot less signal in the middle, resulting in this concave shape. But now if you make the transmembrane mutant, you get still some toxicity because it's not didn't completely eliminate the phenotype. So the expression of GFP is less than GFP alone, but now you don't see this concave shape in the intensity measurements. All right, so just to recap, what I've shown you is I think that DRT2 reveals something really fundamental about molecular biology, and I guess in some that there's another dimension in the genetic code that we would have never discovered had we not gone after understanding the mechanism of these bacterial defense systems. They work by recognizing, the RT recognizes this non-coding RNA and does laps on a portion of this RNA that serves as a template for making a neo-DNA, or nearly endless open reading frame. And that open reading frame then gets transcribed and translated into these neoproteins. The neoproteins are phylogenetically diverse, but all of them seem to rapidly disrupt cell growth. And the way that they're doing that is that they all encode these transmembrane domains. And when we measure ATP concentration in those cells, they dump ATP faster than what you would find in a viral lytic protein. So if you do a control where you express a viral lysis protein, which should explode or disrupt the membrane of the cell, these things work in the same way, but much, much faster. With that, I'll just say thanks to some of our partners who've been a pleasure to work with over the course of the years on these immune systems, in particular David Picard, who coined the term PARIS, which stands for phage anti-restriction induced system, which is appropriate because he's in Paris at the Pasteur Institute. Artem Isaev and Odd Bernheim. The people on the DRT9 work, or the Retron work rather, include Royce Wilkinson, Jaron, and Nate. Murat Bukarik and Royce Wilkinson led the work on DRT2. This is part of the team for DRT9, and I'm ashamed that I don't have a picture of an India and his group up here, but some of the emerging work that we're doing within India and the team here at CIMMYT is spearheaded by an India's team and Mohan now in particular. All right, if you guys happen to know any postdocs that want to move to the mountains of Montana, please feel free to reach out to me. Thanks for your attention.
Thank you, Blake. We all use different tools, including CRISPR, for gene editing and other purposes. So it's nice to see how these tools get discovered and how the new tools are coming up one by one and which can be used for applications in multiple different systems.
Yeah, I think CRISPR's really led the way there, right? But I
Yeah.
think these new systems are new versions with new opportunities, and it's just up to us to be creative enough to figure out how to implement them.
Any question?
I have a question over here.
Yeah.
Thank you very much. It was like an amazing talk. You were telling us about the P retron system that you expressed it into E. coli. Have you think about expressing into plant system or yeast or some other organisms at the moment? Just
We haven't thought about doing that, but it's maybe something worth considering. I guess the question is, why would you do it? And I think there's two possible reasons. There were maybe more, and you could share with me the others. But one is that you make single-stranded DNA. That's what the RT does to create this scaffold, right? So there has been some work, I should say, in human cells where people have used that single-stranded DNA created from the RT as a guide for HDR in genome engineering. So if you make a lot of that multi-copy DNA, you can make it whatever you want. You make that DNA, and now the cell uses it as a template to repair something that's been broken by Cas9, for example. So it's been implemented there, and you could imagine putting it into plants and doing something similar. The other possible way, and maybe you have a different idea again, but this is just something off the cuff, is you could imagine if there's exonucleases in plant viruses, then you could imagine that they would disarm, or rather activate the retron system in a way that would provide defense in a similar way because it would destroy tRNAs. That would mean that the tRNA modification profile in plants is similar to what you find in bacteria. There might be some caveats to think about there, but it's conceivable.
Okay, thank you. And then another question. So you were saying that the type 3 targets RNA, right?
Yeah.
Does this account also for alternative splicing or of there?
Yeah, it can. So you can target whatever sequence you want by design. So if you wanted to target a splice junction that uniquely targeted one transcript that was spliced in a particular way, you could eliminate that transcript and leave the rest of them in their native expression levels. So that would be an interesting application, I think. The real punchline of that paper wasn't that we could target the decimation of RNAs. That had been shown before us, but I simplified the story because really what we showed in that paper was that in the same way that in a human cell, if you use Cas9 to cut the DNA, Cas9 is just the scalpel, right? The two ends of the DNA get put back together by DNA repair systems in the cell, either NHEJ or HDR. But what we showed in that science paper was that the same thing happens with RNA. You cleave the RNA, and the two ends of the RNA get stuck back together, and that activity relies on a host-encoded ligase called RTCB. So the efficiencies of that happening are fractional. I mean, it's a very, very small percentage, but we showed that in a cystic fibrosis mutation from cells that came from patients of a person with CF, that we could target a premature stop codon, excise it from the mRNA transcript. The two ends of the mRNA got put back together and you reconstituted translation. So most genetic diseases are actually caused by premature stop codons, which leads to targeted degradation, nonsense-mediated decay of the mRNA. So you end up with no protein to even treat. You can't develop a small molecule that improves the activity of the defective protein if there is no protein there to begin with. So that's really what we were targeting there. But your question is a good one, and I think that targeting specific isoforms of an mRNA would be a creative application for a technology like this.
Thank you.
Hi, Black. It's a nice presentation, and it's a basic beyond our what we read in the molecular biology. So I have like a question on 3A. what could be the target for RNA virus, particularly in the plant when we are working for the plant system, and it will be a code protein and or a movement protein and or a RNP, so not RNP, sorry, reverse transcriptase. What could be the best target to for the RN3A system?
Yeah, it's a good question. If we're going to target a particular virus, what sequence in that virus would we target and why? And I think one of the nice things about this system that I tried to highlight is that regardless of what you target, it seems to be a little bit target agnostic. It destroys it. So that's like a big win versus, at least my experience, limited experience with RNAi, where every siRNA has different levels of efficiency, none of which are 99% target elimination. Maybe there is some example of that, but that's the exception, not the rule. So which sequence should we target? I guess what I would recommend is that you target a highly conserved RNA that's functionally essential. What you want to avoid is the development of resistance. So if a single nucleotide mutation can result in escape of detection, then that mutation will arise. This system seems to be a little bit less specific, meaning that it could still target most SNPs and eliminate them, but you accumulate a number of SNPs in different locations, each location having a different weight, could result in escape and directed evolution of the virus, which is something that you would obviously want to avoid.
Yeah. So
could I just add one other thing to that? Something that might be worth considering when developing a technology like this is a lot of viruses replicate in so-called viral factories, meaning they set up subcellular organelles where the virus hides out from the host's own immune system, and they replicate inside these viral factories that they set up to hide. So there are different proteins that have to get in and out of these viral factories. So it wouldn't be, it might be appropriate to consider how you would tag the type 3 complexes or components of the type 3 complex to be subcellularly localized to those viral replication centers rather than just floating around in the cytoplasm where they may not see the virus anyway.
Okay, so as you mentioned, non-nuclear decay of premature stop codon.
Yeah.
I have seen some of the paper, even though if, some of a premature stop codon, still we have a transcript. Some transcripts in some example they don't have any transcript, but some example I have seen it has some transcript. What is the logic behind if some has premature?
So if some transcripts escape nonsense mediated decay, I am sure there are exceptions, but as a rule, they usually undergo NMD. But in the case where you end up with these transcripts that don't get degraded, it's probably because the ribosomes stay loaded. And if you have polyribosomes, then the ribosomes protect the RNA from degradation. And then you can result in these N-terminal peptides, some of which are dominant negative. They result in more toxicity themselves. So in that case, it's even more important to destroy the defective RNA because translation of that N-terminal peptide is resulting in some toxicity. In some cases, we understand the function, in others we don't. But being able to repair that transcript and eliminate that toxicity while only repairing a fraction of the mRNA such that the functional protein gets made might be the real sweet spot for a technology like this.
So, in the DR. Okay, yeah.
Any other question from?
Yeah, I have
Go ahead.
2 questions, Blake. And one of them is in CIMMYT and generally globally, many editing systems are still relying heavily on a small subset of nucleases. For us, just as you mentioned, we are mainly using Cas9 and Cas12. And right now we are trying to implement your type 3A system.
Yeah.
But I would be highly interested in knowing, especially based on your expertise about what is available out there, are there any nucleases that you would consider really strong in terms of capability for solving some of the challenges we have in the global south, and you would propose that we need to look at them?
Well, I guess, the largest market share goes to the Cas9 and Cas12, of course. And the reason for that is partly serendipitous, but some of the first nucleases discovered turned out to be the most effective, both effective nucleases, meaning they cleave with high efficiency, they bind sequence specifically, they cleave with high efficiency, and so on. But your question is, are there other hidden nucleases that haven't been repurposed that might address problems in the global south? And I would say it like any tool, the right tool depends on the job. So I think this is where I imagine that the molecular biologist and the plant scientists might come together. You know the problems of the global south. I don't. I'm quite familiar with some of the tools. So maybe by ongoing conversations between the plant experts and the people who know the problems, and by somebody who's familiar with the tools, we can come to the right solution for that particular task. I still hope, I know that you're collectively have been trying very hard to implement the type 3 system, but I still hope that that system might actually prove to be valuable once we can get over some of the technical challenges.
Thank you. I have another question. And yesterday I was having a conversation with an India about an AI nucleus. My former lab has just published a paper. They've been able to use an AI nucleus for prime editing in rice. So how do you see since you've been studying a lot about the natural diversity, but right now there's so much that is coming up on AI nucleases, huh? What do you see as the potential of AI nucleases, and especially for us, considering that the issues around freedom to operate are so intense, and the fact that there is a probability that some of these AI nucleases might be more open access? What's your perception around that area?
Yeah, it's a pretty exciting space. I think this is something that's happening with increasing frequency. I've seen several of these recently. And I think, the power of AI probably can't be overestimated, although some people are trying. But I think with any AI method, you need a good training set. And the natural diversity of CRISPRs that are RNA-guided nucleases, or at least guided nucleases, offer a training set that probably isn't as large as it could be, but sufficiently large now to develop models that can generate bespoke RNA-guided editors that are working with near efficiency of their wild-type counterparts without the benefit of several millennia of evolution to develop these wildly precise homing devices that find sequences inside of a cell. Imagine what they're actually doing. I mean, these nucleases that we kind of take for granted now are basically molecular homing devices that are performing a molecular scavenger hunt, and they evolved with a selective pressure such that if they don't find a target within a few seconds, this virus is going to overtake the cell and kill it. So the selective pressures to develop this efficient mechanism for finding, binding, and destroying a target DNA has been finely tuned over a long, long period of time. And now we can harness all of those years of evolution and put them into these AI models and try to elaborate on that theme, and in a way that maybe eliminates some of the challenges of freedom to operate as you described.
Thank you.
Any other question? I have just one query that there are many viral diseases which create devastation in global south, and the crop we work, maize, wheat, and dryland crops, there are plenty of viral diseases. So we are working on 3A. Easter and Mohan, they started the 3A work, and now Mohan is continuing that. For this DRT2, whether getting something repetitive proteins which could be interesting or useful for cell because it goes in loop, right? So whether that is just getting something in the plant and targeting those against certain diseases or etc. would be the ideal, or you think these kind of many repetitive proteins are of importance for a cell. So should we try to bring those new proteins in the cell system, or you think DRT2 would more work in the line of action against diseases, viruses? Yes.
Yeah, I think it's a really good question and one that we've been pondering for a while in my own lab. I think we have similar questions, whether it's in plants or human cells or wherever we might implement this. There are also some technical challenges to getting this system activated. So something I didn't talk a lot about was the viral triggers that activate these systems and get them to turn on in the first place. If you're going to make a toxic protein, they have to be under very strict control of expression, right? So I'm not trying to dodge your question, but the real answer is I think there's probably applications in targeting disease, but also opportunities for enhancement, both in plant and animal cells that we should be considering. But in order to do that, like at a practical level, you really have to understand how they work. And we're just beginning to understand how they work such that we might be able to engineer them. And that's our ultimate goal. We're not just there yet. It turns out to be more complicated than we anticipated when we jumped into this, but that's part of the excitement as well. It's been frustrating as I don't want to have come across as having two rose-colored glasses on here, but part of the excitement is thinking that you understand how it works, testing that, realizing that you were wrong, and then questioning what is it that we got wrong and why. And I think we're on the cusp of being able to overcome that barrier.
Okay. Thank you, Blake. I will collect all the online questions and send you if I find there are. And thank you all for joining. And thank you, Blake, for coming and give us this talk. And thank you all. Okay.
But this one's better. I think that's right, correct?